Dr. Nardi here, back with another video. Today, we are going to talk about one of the most important ideas in neuroscience, how your brain is built by a collaboration between nature and experience. Think of brain development as a lifelong dialogue between two forces. Nature is the architect. It provides the genetic blueprint, the raw materials, and the rules of construction. Experience is the sculptor. It tests that structure, carving away what is unnecessary and reinforcing what is useful based on your environment. We call this neurodevelopment. Let us look at how the architect starts the process. We start with the very first steps of construction. The entire central nervous system begins as a small patch of cells called the neural plate. If you look at the diagram, you can see the process called neurulation. First, we have the flat neural plate. Then, guided by genetic signals, it folds inward to create the neural groove. Finally, the folds meet and seal to create the neural tube. This tube is the humble beginning of your entire brain and spinal cord. The empty space inside that tube will eventually become the ventricles of your brain. This folding process is high stakes. If the architect's plans go wrong here, the consequences are severe. As you can see in this image of the fetus, if the posterior end of the tube fails to close, it results in spina bifida. If the anterior or front end fails, it leads to anencephaly. But here's our first example of the environment acting on the blueprint. While genetics play a role, a simple environmental factor can intervene. Look at the cereal box on the screen. In 1998, the US mandated folic acid fortification in grains. That one change in the dietary environment reduced these defects by 35%. Once the tube is closed, we need to fill it with cells. Inside the neural tube, we have special cells called radial glial cells. These cells are fascinating because they act as both the factory and the scaffolding. First, they produce new neurons. Then, as you can see in the diagram, the new neurons actually climb up the fibers of the radial glial cells to reach their destination. This creates an inside-out structure. The neurons born first stay deep, and the younger neurons climb past them to the outer surface. If this migration is disrupted, we get conditions like lissencephaly or smooth brain, which leads to severe disability. Now that the neurons are in place, they need to wire up. To form circuits, neurons extend a long cable called an axon. At the tip of the axon is a dynamic structure called the growth cone. Look at how the growth cone is sensing the environment. It is navigating a chemical map. It is attracted to some signals, like neutrons, shown here with the plus signs. It is repelled by others, like semaphorins, shown with the minus signs. This chemical push and pull ensures the axon finds its correct partner. Here is where the strategy shifts. The architect does not build a perfect brain immediately. Instead, it creates an excess of connections. Look at this diagram of a muscle fiber. At birth, shown on the left, a single muscle fiber is wired to multiple motor neurons. This is called polyneuronal innervation. It is redundant and inefficient. Over time, as we move to the right side of the diagram, the system is refined. One neuron wins and the others disconnect, leaving precise control. How does the brain decide which connections stay? It is a literal fight for survival. Target cells release limited amounts of life-sustaining chemicals called neurotrophins. Neurons compete for this chemical. Those that make strong connections get the neurotrophin and survive. Those that are too weak or fail to connect undergo apoptosis or programmed cell death. The architect overbuilds and the environment selects the winners. This leads us to the role of the sculptor, synaptic pruning. If we look at this graph of synapse density, we see a massive spike in connections during infancy and childhood. This is the peak of potential. But look what happens as we move into adolescence. The curve goes down. This is not a loss of function. It is an increase in efficiency. We are pruning away the noise to make the signal clearer. How do we know experience drives this? We look at the classic Hubel and Wiesel experiments on vision. Let me explain these histograms. Each bar represents cells in the visual cortex. In a normal cat, most cells respond to both eyes, represented by the tall bars in the middle. But if you deprive a kitten of vision in one eye during a critical period, look at the middle graph. The brain completely rewires. The cells that would have responded to the closed eye shift their allegiance to the open eye. However, look at the bottom graph. If you do the same thing to an adult cat, the brain does not change. This proves that timing matters. We call these limited time offers critical periods. As this timeline shows, sensory pathways like vision and hearing open and close very early. Language peaks a bit later. If we miss these windows, as happened in the tragic case of Jeannie, who was deprived of language input, the brain creates a permanent deficit. Because the brain is plastic, it is also vulnerable. The sculptor can sometimes be harmful. We know that teratogens like alcohol can disrupt the blueprint, but severe environmental stress like neglect or abuse also physically changes the brain. This MRI scan highlights areas involved in emotion and memory. In children who suffered mistreatment, these specific regions show altered structure. The environment literally reshaped the anatomy. Construction continues with a process called myelination. This is where glial cells wrap axons in fatty insulation to speed up communication. In these scans, the orange areas represent white matter or myelin. You can see that at 5 days old, there is very little. By 1 year, the brain is becoming densely paved with these information superhighways. Interestingly, this is also plastic. Learning a new skill, like juggling or piano, can actually increase a white matter in those specific circuits. This brings us to the final major phase of construction, adolescence. The brain does not finish developing until the mid-20s. The area shown in blue, the prefrontal cortex, acts as the CEO of the brain. It controls impulses and decisions. However, the emotion and reward centers, shown in orange, are already highly active. This creates a mismatch. The gas pedal is fully developed, but the brakes are still under construction. This helps explain the risk-taking behaviors we see in teenagers. So nature provides the blueprint. It gives us the potential. But experience is the sculptor that disposes. From the folding of the neural tube to the pruning of synapses in high school, your brain is a record of your unique interaction with the world. I will see you in the next video.